Hello, my name is Nathan and I'm a neuroscience PhD student at Columbia University. I'm here to introduce you to Late Night Science, a series intended to connect people and science together. We're going to be exploring the stories of people in science and the cool things that they get to do in their labs. If you like what you see, please join us in our in-person events in the New York City area. Details will be after the video. On this edition of Late Night Science, we'll be talking to David Gruskin, an MD PhD student in the Shadlen and Kahn labs. He'll be talking to us about fMRI studies in humans and mice.
I'm David. I'm an MD PhD student here at Columbia, and right now we are in Professor Itamar Kahn's lab at the Jerome L. Green Science Center. So when I was young, about a teenager, my dad got in an accident, and doctors at the hospital that he was staying at used brain imaging to take pictures of his brain and kind of understand how that related to his behavior and his health. And I thought that was super cool. It was a really difficult time for me, but it was really interesting to be able to kind of get distracted by all the bells and whistles of modern medicine and technology. And specifically this idea that somehow using just a bunch of nuts and bolts, we can take pictures of the brain in action and use that to get some sort of understanding of what's going on the inside. So I started college in 2014 at Washington University in St. Louis, where I majored in philosophy, neuroscience, psychology. And that program had us do research experiences very early on. So I started my sophomore year at the medical campus where I worked in a Parkinson's disease lab. And we were using MRI, structural, not functional, to try to predict how people would respond to a certain treatment for Parkinson's disease. So MRI stands for magnetic resonance imaging, and then fMRI is functional magnetic resonance imaging. So that's where we take videos of the brain and get a handle on what the brain is doing and how it's processing information. And that's opposed to structural MRI, which is when we just take pictures of the brain to understand what it looks like, its general shape and form. One thing that I was really interested in studying was how the brain responds to lots of different stuff happening at the same time, which is kind of more similar to realistic, real life situations where people are walking around on the street and you're gonna see a taxi cab drive by, you might hear a bicyclist ring their bell behind you, you're hearing and seeing so many different things at once. So I was really interested in studying movies, which kind of serve as this thing that lights up the entire brain. There's going to be auditory information from the soundtrack and from the voices talking if there's dialogue. There's visual information from the images on the screen. And so there's a lot of stuff happening in the brain when people are processing movies. And the specific project that I initiated was looking at data that was collected actually here in New York from a large sample of kids living in the city who watched a really emotionally evocative clip from the movie Despicable Me. And the idea with this project was that maybe by scanning a bunch of kids while they watched this emotionally evocative movie, we could understand something about emotional disorders like depression, for example. So one of the main motivators for the current project that I'm working on, which I presented at Late Night Science, is the fact that I myself am a twin. So I have a twin sister. She lives across the pond in London, and we have really different tastes in lots of things, and one of those things is movies. So we share a ton of the same genes, about 50%, and we grew up in the same house, but somehow we turned out really different in this way that we enjoy movies. And so that kind of brought me to this general question in biology and neuroscience of nature versus nurture. And in other words, how much of our experience, our subjective experience, and also the way that our bodies work, and here specifically our brains, is determined by our genes, and how much is determined by the environment in which we grow up and develop. And so in this project, I looked at movie watching data that was collected from about 100 twin pairs. So some of them were identical twins, some of them were fraternal twins. And then I could also compare across people in the data set who are not related at all. So take twin A from one twin pair and then look at how they relate to twin B from another twin pair. And the general idea was to see if twins have more similar patterns of brain activity while they watch movies than non-twins. And so looking at the results, that's exactly what I found, which was super cool to be able to put a number on it. A lot of things are heritable, which means that some amount of the variance that we observe in a population in that trait can be attributed to genes. But it's nice to know just exactly how much. And so for this study, I found that the heritability of movie-evoked brain activity, although it differs depending on the brain region you're looking at, is about half as heritable as a psychiatric disorder like depression, for example. So I did maybe 5 or so years of human imaging before I even considered moving to animals. And a lot of the reason that I've made the transition to work with mice is my overarching desire to eventually use the things that I'm doing in the lab to inform clinical care. So fMRI is really cool for understanding how the brain works and may eventually have exciting clinical applications, but one limitation of fMRI is that it's fundamentally correlational. We can take pictures of the brain and we can see how it's processing information while people are engaged in some sort of cognitive task, but we can't say definitively that the areas of the brain that we see light up with fMRI are causally necessary for whatever the person is doing at that moment. So this idea is really important for clinical outcomes and for healthcare research in general, because you want to know if you turn this thing off, say with a drug or say with therapy, for example, is that going to lead to some sort of outcome for this person's symptoms? So one really cool thing we can combine with fMRI in mice is something called optogenetics. So optogenetic fMRI, which is a method that my advisor, Itamar Kahn, played a big role in pioneering about a decade ago, is this method where you use light to activate or silence different parts of the brain. And so while mice are performing a task, we can activate or deactivate parts of the brain to see, number one, if that changes their ability to perform the task or their behavior, and then number two, to see how that affects activity in other areas of the brain. So most behaviors that we're interested in involve lots of different brain regions working together, and if we turn one of them off, we can see how that affects information processing in other areas. I didn't really get introduced to research until I got to WashU, where I started doing that brain imaging stuff. But in that lab at WashU, there was an MD PhD student who was doing really cool research that was very translationally relevant. Ultimately, a lot of the work that we do here at Columbia and in this building is meant to inform the treatment and prevention of disease, but sometimes it's kind of hard to see exactly how to connect those dots if you don't have real-world experience working with patients. And so the MD PhD pathway is one way out of several to get that kind of experience in both domains, where you're working at the bench in your PhD lab and you're also seeing patients at the bedside. And ultimately, best case scenario, you're able to connect the dots between them and use information from both fields to inform how you operate in the other.
Thanks for watching this edition of Late Night Science. Make sure to tune in for our next speaker.